So we're talking about enzyme kinetics, and last time we talked about how enzyme kinetics is one of the most important field when we study enzymes because it studies the speed or the rate of reactions, how fast the reactions go, which is really important because that's what enzymes do. They speed up reactions. And last time we talked about how enzyme kinetics depend on kinetics in general, the kinetics that we studied in Gen Chem 1. These are kinetics that tells us about the reaction order, rate constant, reaction rate, and things like that. So when we look at enzyme kinetics, when we go from general kinetics to enzyme kinetics, one of the things that we saw that is remarkable is that the enzyme acts a little bit different. So instead of having a zero order reaction or first order reaction, what happens is that whenever the enzyme starts, it goes up very quickly and the rate increases as you increase the substrate, which is the first order reaction. The first order reaction increases linearly. And then when the enzymes gets to a certain point, they get to a zero order reaction because they get to a point where the rate is not increasing, the rate is maximum. So we talked about the analogy of a car. When a car starts running, it starts from zero and then it accelerates faster and faster and faster and faster. That is a first order reaction, right? It's going up as it covers the distance. And then when we get to the highway, we get to a point where we have reached the car's maximum speed, where now it's just linear. The more distance we cover, we're not increasing the speed. So this is how the enzymes work as well. We call them first order to zero order kinetics. Now, it used to be very complicated to calculate these orders and these quantities. That is why we switched to something new, which is called Michaelis-Menten kinetics. These are two people who studied the behavior of enzymes, and they came up with new kinetics and new equations to interpret the kinetics of an enzymatic reaction. So how did they do it? Well, they started with this. By the way, Michaelis this lady Michaelis is a legend in biochemistry, and she has a crazy story about how she wanted to study biochemistry from a young age, but she couldn't because at the time women couldn't study biochemistry and they couldn't do science. So she had to go through a lot to get to studying, but she ended up contributing a lot. Michaelis-Menten equation is one of the most important equation in science, and it gave rise to a lot in medicine, and it created a new field of study. So how did they do it? It was Michaelis and Menten, and they looked at this equation. So not only do we have a two-order reaction, we also have two different reactions. We have an enzyme plus substrate that gives us an enzyme substrate complex. This is one reaction, and then another reaction is the enzyme substrate complex gives our enzyme plus product, right? So we have a lot of moving pieces. We have enzyme, we have substrate, we have enzyme substrate complex, and then we have the product. And then at every equation we have rate constant. So you have the rate for the reaction, but also you have the reverse rate, because most of the time the reverse reaction is also true. And then you have the second rate. So how do you combine all of this, combined to the fact that we have the first and the first and zero order kinetics, to get to a quantifiable reaction? Now, if this was in grad school, we would have to work out to how they derived it and how they got to it. But I'm just going to show you the reaction. The equation gave us that the rate of the reaction is equal to Vmax times the substrate concentration over Km plus the substrate concentration. What's Vmax? What's Km? Well, let's look at the enzymatic reaction that I showed you earlier. So if we have a reaction like this, right, and then our enzyme is going to start like this, and then it's just going to level off, right? And then we measuring the substrate concentration here, and then we're measuring the rate here in V. Well, the maximum velocity that the enzyme has gotten to is called Vmax. Easy, right? This is what we call Vmax. And then when we get to half of Vmax, and they go this way, this is Km, a very important value. Is the substrate concentration corresponding to half Vmax. So this comes from this equation. Rate is equal to, again, it's Vmax times the substrate concentration over Km plus the substrate concentration, right? So this equation is represented by this graph. This is the Michaelis-Menten kinetics. This is the graph, Michaelis-Menten graph. This is Michaelis-Menten equation. And now we know all of these meanings. And what it really means, if you remember your mathematics, it means that for each velocity point here, you can calculate its corresponding s. So for each velocity point, you have the corresponding s. So these are what we call our variables, the things that change as the graph changes, right? Think of x and y, but this is v and s, right? But also you have the Vmax and Km. These are our constants. So in this equation, you have constants, you have variables. Vmax and concentration of S are variables over time. And then, the velocity and concentration of S, the variables. Vmax and Km, the constants. And that is the Michaelis-Menten equation. A little bit about Km. The Km is a constant, and it is derived from rate constants. So going back to the Michaelis-Menten, if we were to derive it ourselves, when you setting up the equations, you get to a point where you have this right here, and then this is what we call Km, right? So this is where it's derived from, but it is very, again, the significance of it on the graph is this, right? It corresponds to half of Vmax, but also it shows the affinity. So a lower Vmax shows a high affinity of the enzyme. Not lower Vmax, low Km, my bad. Low Km shows high affinity. High Km shows low affinity. All right, so that means that if we go back here, if we have another enzyme that looks like this, that goes this way, right? Look, it's not as good, right? So this is its Vmax. Half of its Vmax will be here, so the Km will be bigger. The Km will be bigger. So the Km for this second enzyme will be bigger. And this enzyme is not as good. How is it not as good? It doesn't go to the Vmax as quickly, right? So this is enzyme affinity, right? So Km can show you the enzyme affinity. Vmax is also a constant. One thing to note is that most of the Vmax calculations are theoretical because we can never achieve full Vmax, because to reach it, it will mean that all of the enzyme have bound to all of the substrate, and that's not just possible in a moving dynamic environment. All right, so a few other terms that you will see in enzyme kinetics related to Michaelis-Menten: Kcat. Kcat is the turnover number. The turnover number or just catalytic activity. And it's very important. It tells you how many molecules, how many substrate molecules are converted into products per enzyme per unit of time. Right? How many molecules of substrate are turned into the product per enzyme per unit of time. That is Kcat, catalytic activity or turnover number. And it's most of the time in the Michaelis-Menten model, Kcat is also equal to K2 or just Vmax over the enzyme total concentration. Remember, K2 is here, right? K2 is this second rate, right? So that's the turnover number or catalytic activity. Enzymes have turnover numbers from 40 million per second. If you can imagine 40 million per second, that's a lot of molecules. It's even hard to imagine that an enzyme can do that. But also you have slower enzymes that have even less than 1 molecules per second. So enzymes vary a lot. But it's not just based on how good an enzyme is. It's also based on how complex the reaction is. All right, another one that I want to talk about is called the catalytic efficiency. Catalytic efficiency kind of shows you how perfect an enzyme is, how good it is. Not just so Kcat measures how many molecules we're catalyzing, right? And then Km measures the affinity, how quickly it binds to it. So Kcat over Km is the catalytic efficiency, right? That's another one. And it also, by the way, sometimes it's called KEF. That's another K that we need to know. KEF, that's the catalytic efficiency. So we have Kcat, Km, KEF, and we also have K1, K negative 1, and K2. And K1 and K negative 1, by the way, sometimes are called K on and K off. It's the same thing, really. But we just need to know all our Ks when it comes to enzyme kinetics, right? And these are examples of some catalytic efficiency for some enzymes. So this equation right here, right, the Michaelis-Menten equation, there are so many things that we can do with it. But in biochemistry, you encounter a lot of problems asking you to either calculate V when you're given Vmax and S or Km. A trick question that I might ask you is, hey, what is V, right? Yes, what is V when Km, no, what is V? Oh, that's it. I'm trying to find a trick question. Oh yes, what is V when S is equal to Km? What is V when S is equal to Km? Well, when S is equal to Km, when S is at Km, V is equal to V max. That is the definition, right? The definition of V max is when S is at Km. So, no, half of V max at least. Okay, I'm gonna have to write down this trick question because it's tricking me. But basically, it's asking you like, hey, what is V when S is equal to Km? And V is going to be equal to V max over 2. Yes, that's exactly it. It's going to be equal to V max over 2. Let's look at another question that is written. Okay, so if Vmax is equal to 3 millimolar per second for a Michaelis Menten enzymes, what is the velocity when S is equal to Km and is equal to 10 millimolar? So this should be just an easy plug-in. So what is the velocity, which is V, which is the rate, is equal to Vmax, which is 3 millimolar per second, times S, which is 10 millimolar, over Km, which is 10 millimolar, plus S, which is also 10 millimolar in this case. This one is pretty easy. So it's going to be equal to 30 over 20 basically. And the velocity is in millimolar per second, and this is just 1.5. That's too easy, right? Just plug in and play, no problem. Let's see another question. And I added the equation here, by the way, but it doesn't mean that it's going to be given to you in a question, right? It's something that you're gonna have to know. All right, so this one says, if an enzyme has a Vmax of 15 millimolar, what is the velocity if the substrate is present at half of the Km? So they gave us the Vmax. That's good. They didn't give us the Km, they didn't give us the substrate, but they tell us that the velocity, they tell us the substrate is equal to 1/2 of Km. This is very good information, by the way. This is a little bit of algebra. So all we gotta do is just express one in terms of the other so that they can cancel out. What do we mean by that? I'm just going to do Km because it's easier, but here 2s is equal to Km, right? So just cross multiplication. So I'm just going to do the equation, but I'm just going to replace Km by 2s because that's what the question says. So I'm just going to say V is equal to V max, which is 15 millimolar per minute, times ACE, and look at this, over Km is equal to 2S, so over 2S plus S. And look at this, when we combine the S's, they are going to cancel. So here V is equal to, let's remove the units real quick so that the things are easier. 15S over 2S plus 1S is equal to 3S. So this is just going to be equal to, so the S's are going to cancel, and we're just gonna be left with 5 millimeter per second. Does that make sense? You can go ahead and rewind that if you need to understand that a little bit better. But that is what we do. That's how it goes when it comes to solving Michaelis-Menten related questions. So all you gotta do is just remember this equation and just be more comfortable moving this around. Now here is another question that I like to ask, and a lot of other people like to ask it too, and it's not just because it might be tricky, but it's because this is how we do it in the lab. So in the lab, you can measure the velocity and you have the substrate concentration. You can actually plot this. You can plot this so that you can from your graph calculate Vmax and Km and things like that. But there is a better way to do that. What's a better way to do that? It needs introduction of another equation. It's not another equation, it's kind of a turn, a spin, if you will, of Michaelis-Menten equation, and it's called the Lineweaver-Burke equation. Lineweaver-Burke is these two people who elaborated on the Michaelis-Menten equation. And all it is really is just the reverse of the Michaelis-Menten equation. What do I mean by that? So let's put it on a new layout, and I'm just gonna bring in the equation. There you go. So if you remember, the Michaelis-Menten equation is like this, right? It's V is equal to Vmax S over Km plus ACE. So I promise you this is how it is. If you take this, and we can do that pretty quickly, I think we can do that. If we reverse this, 1 over V is going to be equal to, it's a reverse of this, so it's the reverse of Km plus ACE over Vmax times ACE. And since there's a plus here, we can divide this into two fractions, right? So it's Km over Vmax S plus S over V max S. And look at this, because the S's are going to cancel, then it gives us this equation on top right here. And that is called the Lineweaver-Burke equation. So again, it is 1 over V is equal to Km over V max times 1 over S plus 1 over V max. Now, why do we care about this? Why do we care about this so much? This is because this equation is going to give us a linear plot rather than a curve. All right. So if we were to estimate, going back to the Michaelis-Menten, if we were to estimate the Vmax, it's very hard to do it in a curve like this, or even the Km. But if they were on a linear graph, it will be much easier. So it turns out that, if I can draw this, well, so it turns out that if you plot the variables from this equation, and if you can tell, again, the variables from this equation are 1 over V here, that's our Y, and then 1 over S here. If you plot this, then instead of having a sigmoidal shape like the Michaelis-Menten, this one is going to be a straight line. It's going to be a straight line like this. Okay, this is not very straight. A little bit better, but it's going to be a straight line. And with a straight line, you can then calculate a few things from there, right? You can calculate the variables, the variables that we're trying to calculate much easier. So with this 1 over V, look at this, the y-intercept, if you remember a little algebra, if you take this whole thing, right, if this, you take this whole thing as y is equal to mx plus b, then b is going to be the y-intercept, and the y-intercept is in here. Look, it's going to be 1 over V max. So the y-intercept that you can calculate easily is going to be equal to 1 over V max, right? That's pretty cool, right? So if you do this and you have a number here, is going to be equal to 1 over V max, and you can easily calculate V max from there. That's why the Lineweaver-Burke equation is much easier. So another one here, right, the x-intercept, if you remember from your algebra, m here is the x-intercept, right? So the x-intercept for this one, the x-intercept is going to be here equal to negative 1 over km. So with the Lineweaver-Burke equation, if you plot it, you can easily calculate Km and Vmax. So for example, if we do an experiment, right? Let's say we run an experiment, and then we get we have the rate, and then we have the substrate, and then we plot 1 over substrate versus the 1 over rate, and then this line gives me an equation. So remember, in Excel you can plot this and then just get a line. Just get an equation for your line. So if this line says that, oh, okay, this line is V. So Excel is going to give you like y is equal to 2.5x plus 12. If this is what the line is equal to, if your data says this, that means that this 12 is equal to 1 over max. So your data will be 1 over max, Vmax, is equal to 12. And then Vmax is going to be equal to 1 over 12, right? And then here, same thing, if this is 2.5, here will be negative 1 over Km, and then you can find Km from there. So this is an important tool to calculate both the Km and the Vmax from experiments, right? So even though the Michaelis-Menten equation is very important, and we use that in calculations and things like that, but in experimentation, we take it further and make it linear by using the Lineweaver-Burke equation, right? Because the plot for this is going to yield a straight line, whereas the Michaelis-Menten plot is a curve that's hard to calculate parameters from. All right. I think that is a good overview on calculations involving kinetics. Now, enzymes are also dependent on pH and temperature. So there are many things that affect enzyme function. But some of the external things are pH and temperature. So most of the enzymes are going to be working in a defined acceptable range of pH and temperature. Now, when it comes to temperature, let's start with pH actually. So pH affects not only enzyme catalysis, but also enzyme recognition. The enzyme recognizing the substrate. That is a very important point to make, because remember that an enzyme has an active site, and the active site is lined up by amino acid side chains that are going to recognize the substrate. So if you change the pH, guess what happens to the side chains? They're going to change, right? If you remember, some of them are going to be deprotonated if we go above their pKa's, right? So if, for example, it was going to be a group that's acidic, and then it's deprotonated, now it's no longer acidic, is it? So it's not going to bind to that substrate. So if you change the pH, the substrate recognition is going to change, and the catalysis is going to change as well, right? And again, it's because of the ionizable side chains in the active site, and also it's the ionizable side chains in the enzymes as well. Like outside the enzymes, if you change the side chains, the enzyme is also going to undergo a structural change as well. That's going to change the size and shape of the the active site, right? So the substrate also may change, the pH may also change the substrate. So the effects of pH can and they do affect Km and or Vmax, right? So a lot of enzymes are going to have a pH at which they are comfortable. And guess what? A lot of enzymes are comfortable at physiological pH, right? Between, as you can see here, between 6.5 and 8.5, but there are some enzymes that are comfortable at very low pH and very acidic conditions. Can you guess what those are? Stomach enzymes, right? Stomach enzymes like really low pH. But many other enzymes are usually comfortable in plus or minus physiological conditions, all right? Now, in terms of temperature, it's a little tricky. Why? Because generally speaking, if you increase the temperature, the enzyme is going to work better, right? If you increase the temperature, the enzyme is going to work better. In fact, if temperature is too low, enzyme is not going to work. So most enzymes work well in temperatures between 50 to 60 degrees Celsius, right? No. That's actually too high. Most enzymes work well around the physiological temperature. Does anybody know what the physiological temperature is, by the way? It's going to be around 37 degrees. So enzymes work well around that. But if you make them low, if you make the temperature lower, the enzymes are not going to work. If you increase the temperature a little bit, the enzymes are actually going to work better, but not forever. You're going to hit a temperature where the enzyme is going to break down, right? So we talked about this when we were talking about proteins, but at a certain temperature, a protein is going to unfold and lose its tertiary structure and even secondary structure in some cases, and just be a single ribbon. At that point, the enzyme is not going to be functional, right? So temperature, increasing temperature increases enzymatic activity up to a point, right? So usually a general rule is that the enzyme rate doubles every 10 degrees Celsius, but only in that window of acceptable temperature, right? If you go above that, it won't work, right? So this is an example of what happens. You increase the temperature, the activity increases until you hit the high temperature, and then it starts to decrease, right? So for a lot of the enzymes and proteins that we work with, these are very common graphs that we do. Just vary temperature is a very easy experiment to do in the lab. So you vary temperature, you have different temperature points, and then you measure enzymatic activity, and then you just plot it. You see how it goes up and then how it goes down. Same with pH. It's a very easy experiment to do. Now, there are other things that naturally affect enzymatic activity, and these are what we call regulators or enzyme regulation. So there are a few of them, right? The best way to regulate enzymatic activity is a mode of production of that enzyme only when the substrate is present. So when the presence of the substrate triggers the production of the enzyme. That's the best way to regulate it, but usually it doesn't happen in humans, but it's common in bacteria. The second one is allosteric enzymes. What are allosteric enzymes? These are enzymes, and I want everybody to understand. So Usually when you have an enzyme, let me open a new window. Usually when you have an enzyme like this, you have an active site, right? You have an active site where your substrate binds, right? So this is the substrate. This is the active site. All right. So in a lot of enzymes that we call allosteric enzymes, they have a site away from the active site that we call an allosteric site. And this site right here can be an on and off switch for the enzyme. So we can bind things here that are called regulators. So we can add things that are called activators or repressors and things like that, and inhibitors that are going to bind away from the active site. So this is what we call an allosteric enzyme. So this is one of the ways enzymes are regulated. So that's allosteric inhibition. Feedback inhibition is when you made too much of a product, right? When you made too much of a final product and it comes back to inhibit the initial enzyme, or one of these. So feedback inhibition is like, hey, we made too much, stop. And this happens a lot. This happens a lot in glycolysis and glucogen metabolism and glucose metabolism, where you made too much of ATP, for example, and ATP comes back to inhibit enzymes in the glycolytic pathway because it's like, hey, we've made too much ATP, slow down. This is called feedback inhibition. All right. All right, what's another way to regulate enzymes? Zymogens. Zymogens are immature versions of proteins. That's the best way to put it. It's a protein it's an enzyme that is not fully formed. Something has to happen to it for it to start to work. So an example is, for example, trypsinogen versus trypsin, right? Trypsinogen is a protein, but it's not active. It needs to be cleaved into two parts for it to produce trypsin. It needs to be, you need a peptidase. You need an enzyme that comes in and actually cleaves it, and then it becomes active. That is what we call a zymogen, right? Protein modification is another very powerful way to regulate enzymes. An example, a very common, powerful example that the cell uses is phosphorylation. So you're gonna have a lot of times an enzyme that is not active, but when it's phosphorylated, it becomes active, right? or vice versa. It's inactive, but when it's phosphorylated, it becomes active. So phosphorylation, and we have three amino acids that can be phosphorylated, and we're going to talk more about that. But this is another powerful way. So we have phosphorylation, we have acetylation, you have methylation, and other ways to control and regulate enzymatic activity. Right. Now, another way that's not here because we're not talking about regulation, but another way to affect enzymatic activity is inhibitors, right? Inhibitors or inhibition. So how do we inhibit enzymes, and what type of inhibitors do we have? Well, we have irreversible inhibitors and reversible inhibitors, right? So we can classify enzyme based on reversibility or competition. All right. So reversibility, you have reversible and irreversible inhibitors, right? An inhibitor can be reversible or irreversible. Reversible means that the inhibitor can bind, inhibit the enzyme, but then leave after. Irreversible binds to the enzyme and then it's bound to it completely. And this is usually by a covalent bond, right? Reversible inhibitors are bound by weak bonds like hydrogen bonds or electrostatic interaction. Irreversible inhibitors are usually bound by a strong bond such as covalent bond. Right? So reversible, irreversible when it comes to reversibility. But when it comes to competition, you have three, right? So you have competitive inhibitors, non-competitive inhibitors, and uncompetitive inhibitors. All right, so those are three inhibitors. So again, reversible inhibitors bind to an enzyme and then they can leave. Irreversible inhibitors bind to an enzyme and they are bound forever. That's reversibility. Competition, when an enzyme, so you have an enzyme, you have the active site here, right? When an enzyme binds to a substrate here, to the active site, and then you have an inhibitor that comes and competes with the active site, competes with the substrate at the active site, that's a competitive inhibitor. A competitive inhibitor comes in and binds to the active site. That's a competitive inhibitor. And it can be reversible or irreversible. And then you have other enzymes that instead of binding to the active site, other inhibitors, instead of binding to the active site, they can come and be like, bind here, bind outside the active site, but still change the enzyme in a way that it doesn't work. Those are non-competitive inhibitors. Non-competitive inhibitors don't compete with the substrate. They still bind to the enzyme, change its structure to the point where it doesn't bind to the substrate, but they don't compete with the substrate, right? That's non-competitive. And then uncompetitive inhibitors. A lot of people neglect these, but these are important. Uncompetitive inhibitors bind to the enzyme substrate complex and keep it there, right? So remember, the enzyme substrate complex produces our product. So you have you have enzyme, you have substrate, they bind together, you have the enzyme substrate complex, and then they go through a change, and then you have on the other end enzyme and product. So uncompetitive inhibitors bind to the enzyme substrate complex and they lock it there so that it doesn't produce our product. That's uncompetitive inhibitor. Very important distinction. I want you guys to note these three, okay? And again, these inhibitors, reversible or they can be reversible or irreversible, okay? But most of them, I'll say, are reversible, okay? I have to be clear about that. Most of the inhibitors are reversible. And that's what we want, right? Because we want our enzyme to be reusable, right? We don't want to get rid of our enzyme. So most of the inhibitors are reversible. So that's how we rank inhibitors based on reversibility and competition, okay? Now, again, I talked about three types of inhibitors, right? Three types of inhibitors when it comes to competition. You have competitive, non-competitive, and uncompetitive. Now, what if I told you that we need to know and we have to know how these relate or affect our kinetics? In fact, we have to know how these affect our Michaelis-Menten plots or our Lineweaver-Burk plots. And you might think that that is difficult, but it's not difficult at all. You just need to know that all of these affect either our Km, our Vmax, or both. And some of them make them go up, some of them make them go down. So let me show you something that kind of puts all of it together. So look at this. You see, the top is the Michaelis-Menten graph. Let me move this here. And the bottom is the Lineweaver-Burk plot. And look at how, right? The solid line is the uninhibited one, right? So look at how, okay, you start with a nice plot. But if you have a competitive inhibitor, what is it going to do? It's still going to reach the Vmax, but the Km is going to be affected. The Km is going to be higher. So for a competitive inhibitor, you have Vmax stays the same, Km increases. And if you remember what Km means, the increase of Km means that the enzyme has less affinity, right? All right, let's go to non-competitive. Non-competitive inhibitors, look at what happens. Km stays the same, Km doesn't change, but Vmax decreases. So Vmax decreases, Km is unchanged. That is a hallmark. That is the biggest difference between the two. Vmax in competitive inhibitor, Vmax is unchanged, Km is increased, and then in non-competitive, Vmax is decreased, Km is unchanged. Right? And look at this. In uncompetitive inhibitors, both Vmax and Km are decreased in uncompetitive inhibitors, right? And now look at the bottom. This is how this is represented, right? In the linear form, remember Vmax is the intercept, right? So what ends up happening is that a competitive inhibitor is going to go through the same line but end up with a different number here, right? What about a non-competitive? Non-competitive, the intercept at Km is going to be the same, but the Vmax is going to be decreased, right? Remember, you might think, oh, it's increased, but this is 1 over Vmax. 1 over Vmax increases, but that means that Vmax decreases, right? And then when you have uncompetitive, both of them decrease, so it moves this way, right? So why is this important? It's because if I have an inhibitor that I've never tried on my enzymes, guess what happens? I'm going to study it, and I will not only find the Km, so I will find the Km and the Vmax of the enzyme by itself, then I will find the Km and the Vmax of the inhibitor, and then I will see, oh, okay, based on these changes, is my inhibitor competitive, non-competitive, or uncompetitive? So this is something that we do in the lab, by the way. We look at these numbers to learn what type of inhibitor that we have in our test tube, right? Now, I'll tell you right now that sometimes some of them won't fall into any of these. And we'll just say that it is a mixed inhibitor. It's a mixed inhibitor. So it would mean that maybe it's uncompetitive and non-competitive mixed. And it can be as complex, but when that happens, you have to repeat your experiment to make sure that you still get the same data. And if it's the same data, then it's a mixed inhibitor. But usually they should fall into either competitive, non-competitive, or uncompetitive. All right. So if I give you data, you should be able to plot it. And then if I add inhibitor data, you should be able to tell me what type of inhibitor it is. And you should be able to use these to find Km and Vmax and all that stuff. All right. So this is why enzyme kinetics is fun and also very useful in the lab, because you can actually use it to test actual potential drug candidates, and these are what they use to actually discover drugs. Now, some of them are a little bit more complex than that, but the idea is still the same. All right, so I think that's enough about enzyme kinetics and enzyme regulation. But with this video, I think we've been able to look at an overview of enzyme kinetics, look at some of the calculations that you can do using enzyme kinetics, especially the Michaelis-Menten equation. That equation is very important. I hope you've already memorized it. If you haven't, make sure that you do, because you're gonna need it. But also we saw how we transform that equation into a linear equation, and from there you can calculate Km, Vmax, and things like that. We looked at a few ways to affect enzyme activity. So actually this is a good question that I can ask, like, hey, what are 10 ways we can affect enzymatic activity, right? And you might think like, oh, we've only talked about 2 that are pH and temperature, but no, not just that, right? We looked at pH, we looked at temperature, we looked at inhibitors, different, 3 types of inhibitors. We looked at allosteric regulation, we looked at feedback inhibition. So we looked at many different ways that enzymes are affected, either in the lab, outside the lab, in the body. And we looked at different, we looked at how enzymes function at physiological pH and physiological temperature, and what happens when we increase the temperature or increase or decrease the pH and things like that. So make sure that you take that as a take-home message, that there are so many things affecting enzymes, but we cannot only learn how they affect that, but we can calculate it. We can calculate it to the very to the very number of how much they affect, for example, our enzyme activity. We can look at inhibition and things like that. So this is a good video, and I think this is a good place to end. Please send me a message if you have any questions, and I'll see you next time. Bye bye.